So today what I'd like to do is I've broken this up into 2 sessions. So I know we have 2 hours here, so hopefully I don't put everybody to sleep, but we'll take a little break in between halfway through. So in the first session, what I'd like to talk about is a quick review about the methods that we have for 3D imaging, or the methods that we had prior to Lightsheet. Introduction to what a Lightsheet microscope is and a little bit of the history behind it. We'll talk about how you generate a Lightsheet and the different varieties of Lightsheet microscopes that are out there, and some of the appropriate sample types for this type of microscopy. And finally, a little tough situation that we come into contact with Lightsheet is data handling. So we're going to touch on that as well. And then we'll take a bit of a break, and we'll come back, and after the break, we'll talk a bit about sample prep. And I'm going to talk specifically for a multi-view Lightsheet setup, sample prep for that, which is what the Zeiss system is that I believe you guys are currently looking at. And then we'll also talk a bit about tissue clearing. Okay. So in the past, fluorescence microscopy has definitely arisen as one of the best techniques we have for generating contrast in a sample and also having molecular specificity. So we're able to look at specific molecules in our sample with high resolution and high contrast. And the way we've done that is primarily through 2 microscope techniques. One is your standard wide field microscope. So a very simple light path. We have some white light that comes in. We have some filters to send some excitation light up onto our sample. We get some fluorescence that comes back into our objective and goes to our detector. Of course, this works really well for nice thin samples, but once you get a thick sample, we all know you end up with a very blurry image. And so the advent of the scanning confocal microscope changed this light path around a little bit in that our laser beam now, this is actually poorly drawn. It shouldn't be focused into the back of the objective here. It should actually fill the entire back of the objective. Which means you focus your light, your excitation light, into a single point in your sample. You get your fluorescence, you collect that fluorescence, send it back, and the key component right here is this pinhole that sits in front of our detector and excludes all of the fluorescence light coming from above and below our focal plane. And then this is what allows us to take these nice optical sections, these thin slices through our sample, and build those up into a 3D volume, similar to what you do with a CT scan or an MRI. All right? But there's a couple problems with these designs. One of them, you can see this excitation light, this blue light, is going all the way through our sample in both of these situations. And so what that means is we're doing photo damage and bleaching outside of our focal plane. With this design here, we're focusing just at a tiny point in our sample, which means we have to scan that back and forth all across our sample. This ends up being pretty slow. All right? So these are 2 of the big limitations. With wide field, or 3 big limitations. We don't have optical sectioning. Over here, it's really slow to build up that optically sectioned 3D volume. And in both samples, we're doing photo damage and photo bleaching all the way through our sample. Okay? So that's just outlined here. So with the wide field microscope, we have the disadvantage of no optical sectioning. With a point scanning confocal, it's really slow. And with both of these designs, we have light exposure through the sample. We have a single optical axis, which often means we have limited light penetration if we have a really thick sample or a really scattering sample. And to some extent, we also have some difficult environmental control. So prior to Lightsheet, I guess this shouldn't say current, it should say prior to Lightsheet, there were a few solutions or proposed solutions to get around these issues. So we've probably all heard of spinning disk microscopes, where we basically parallelize our excitation light. So instead of just focusing on a single point, we have this spinning disk with a number of openings in it where we can focus multiple points of light into our sample at a time. So we're parallelizing that acquisition. So this ends up being much faster. We also still have optical sectioning here. But we have the disadvantages. We still have that light exposure all the way through our sample and limited penetration. 2-photon microscopy gets away from this excitation all the way through our sample. So this is kind of the famous picture of 2-photon microscopy. You can see on the left here we have a standard one photon excitation process where we have that excitation light passing all the way through our sample. But over here with the 2 photon excitation, it's tough to see, but there's just a tiny little spot right there. And that's the only spot where excitation is happening. So we only get excitation at our focal plane. We get improved light penetration because we're using infrared light, which penetrates better through biological tissue. But once again, we're back to a really slow point scanning process where we're having to scan that laser beam back and forth across our sample. All right. So we've addressed a couple of the issues, but we still don't have a design that addresses all of the problems that we find with microscopy. So the question remains, can we build a microscope that can optically section so that we can build up 3D volumes? Can it image a large field of view? Can it only illuminate at the focal plane so that we avoid doing photo bleaching or photo damage to our sample? Has some good environmental control and has deeper sample penetration. And the answer to this is actually to look back into history. So Lightsheet microscopy is also a Nobel Prize winning technology. The Lightsheet microscope was actually designed way back in the early 1900s, won the Nobel Prize in 1925. But obviously, we didn't have fancy things like lasers back then, and this was designed to look at gold colloids in solution. So Zsigmondy designed this microscope where he had a little cuvette here that sat with his gold colloids on it, with the standard microscope imaging axis that everyone's used to. But then he came in with a second lens group here on the side that focused excitation light. He had a light source over here into his colloids. Right? So the design has been around for many, many years. The first modern update to this came 90 years later. So this was published out of the Spellman Group in 1993. And essentially, this is the same idea. So you have a couple lasers now that are coupled in and go through what's called a cylindrical lens. We'll talk about this in a little bit. Form a narrow sheet of light going through your sample. And then there's an imaging objective up here and a camera that captures that fluorescence coming out of your sample to image it. And you can move the sample back and forth through this light sheet and take optical sections throughout your sample. All right. So that was the first modern version of it. And as we always do in microscopy, we try to challenge the signaling world to see if we can come up with more acronyms than they have. And quickly there became this alphabet soup of all these different light sheet techniques. Now, unlike the super resolution field, all of these light sheet people got together and decided they were going to come up with one term to describe this. So they came up with the term light sheet fluorescence microscopy, or LSFM. So I think this has been both an aid and a hindrance, because now everything's called the light sheet microscope, but there are definitely many different designs within the light sheet umbrella. So I think for the most part, they can be grouped into these 3 different categories. So beam scanning designs, multi-view designs, and then those that are based on a stereo or zoom microscope design. All right? So I'm going to come back to these in a little bit. But first, what I want to talk to you about is resolution in the light sheet microscope. So this is usually one of the very first questions anyone asks me when they want to come and use a light sheet is, what kind of resolution am I going to get? How is this going to compare to confocal? How is this going to compare to wide field? So hopefully you're familiar with a point spread function. So what this is just a theoretical model of what an infinitely small point of light that's sitting in your focal plane looks like when it gets back to your detector. So due to the wave nature of light, we have a lot of diffraction as it passes through the optical components of our microscope. And an infinitely small point, once it gets back to our detector, actually looks like this blurry spot with a number of rings around it. Okay? I've actually enhanced the contrast here so that you can see the rings. They aren't quite that bright in reality. So this is a theoretical point spread function in the x and y plane for an objective with a numerical aperture of 1.0, with a sample sitting in water with a refractive index of 1.33, and we're looking at orangey red light around 600 nanometers. Okay? So this point spread function in x and y, you can see here we have, if we just draw a line through it and do an intensity profile, we have a full width half maximum of about 350 nanometers. So that's approximately the resolution of the system. This is defined by this famous equation by Ernst Abbe, who used to work for Zeiss. That just says that the size of the spot is basically dependent on the wavelength of light that you're looking at and the numerical aperture of the objective you're using. So like I said, this is in the XY plane. And this is pretty much identical whether you're using a confocal, wide field, or a light sheet. Okay, so there's no difference in the XY resolution with a light sheet. The big difference is in the Z dimension, or the Z dimension. I've totally become an American. So if we look in the Z dimension, our point spread function looks much, much different. So this is an XZ projection of a point spread function. And so here it's the exact same conditions, the same numerical aperture, same refractive index, same wavelength. Again, I've enhanced the contrast so you can see some of these side lobes around the central focal point. But what you can see is the equation that we use to define our resolution in Z is a lot different. And this ends up giving us a much larger point spread function and lower resolution in Z. Okay? So this actually works out to a full width half maximum of 1.6 microns. Okay? So the difference with a light sheet is we're actually dealing with two point spread functions. So this is what the inside of the Zeiss light sheet microscope looks like. So you have two objectives on the side here that are projecting in your light sheet, and then you've got an objective at the back that's collecting your fluorescence. And we'll come back to this again in a bit. But if you're looking down on the very top of this chamber, here's what you see, your excitation objective on the side projecting in your light sheet, and an objective up on the top here that's collecting your fluorescence. So I've just shown this projecting in some green light, collecting some red fluorescence. And of course, these objectives here that are projecting in your light sheet have a point spread function, and this objective here that's collecting your fluorescence has a point spread function as well. So the effective point spread function in Z of a light sheet microscope is actually a convolution of these two point spread functions. Okay? And this can have two very different effects depending on what your imaging objective, the one that's collecting your fluorescence light, which type of objective you're using. And the big difference is whether you're using a high numerical aperture objective, so this is a 1.0 water dipping objective, or a 5x air objective with a low NA, so this is just a 0.16 NA. All right? And the big difference here is the depth of field of these objectives, or the length of that point spread function in Z, is much, much different. If you remember, that point spread function in Z is dependent on the square of the numerical aperture. So here with this low NA objective, we have a depth of field that's greater than 40 microns in size. So it's really big, very, very poor Z resolution. With our 20x objective, we've got that depth of field that's less than 2 microns in size. So if we go back to this diagram again, and we plug in point spread functions for these imaging objectives, or the ones that are collecting the fluorescence, that are relative to their actual numerical apertures, what we see here is we have our light sheet coming out of this objective, and it's going to be the same in both of these examples, but our point spread function of this imaging objective is much different. Okay? So this is actually not even the full point spread function. I've chopped it off just to make it fit in the image. But you can see this one transcends a much wider dimension than the actual light sheet itself, whereas the point spread function for the high numerical objective is actually confined within that light sheet. So what this means is that when you're using a low numerical aperture imaging objective to collect your fluorescence, your light sheet is actually determining the thickness of that optical section, or your Z resolution. Whereas with a high numerical aperture objective, it's actually the objective itself that is determining the optical section. All right? So in this situation, a light sheet microscope actually has a better Z resolution than a confocal. In this example over here is actually quite comparable to wide field. The one exception to that is, in most cases, your sample is actually going to be thicker than the thickness of this light sheet. And so you are excluding a lot of out-of-focus light that you would still get with a wide field system if you had that thick sample. So I say comparable, but if you have a thick sample, it's still a better contrast because you're removing some of that out-of-focus light or that blur. All right, so just to beat the dead horse one more time, if we actually take a look at this convolution of these two point spread functions on top of each other, so for our low numerical aperture objective, here's what our point spread function looks like for that objective. I haven't shown you the light sheet point spread function, but it would be lying across this one in this dimension. And here is our effective point spread function now. Okay? And the exact same thing for our high numerical aperture objective. So here's the standard point spread function. Our light sheet would be coming across here. And here's our effective point spread function in the end. Okay? And here our full width half maximum is determined by the width of the light sheet, where over here it's the point spread function of the imaging objective. All right? Does that make sense? Okay. So how do we actually make a light sheet? So there's a number of different ways that this has been done. The sort of classical method is to use what's called a cylindrical lens. I'll show you this on the next slide. Essentially what it does is just focuses light in one dimension so that you still get this nice wide sheet, but it's nice and thin in this dimension here. Okay? And that just forms a sheet right through your sample that you can place your sample in. The other option is to take a Gaussian beam. So this is exactly what's coming out of your confocal as you're using it right now. And instead of raster scanning that back and forth across your sample, you actually just scan it in one dimension back and forth in a line. If you scan it back and forth really, really quickly, so that you're covering your sample multiple times during the exposure time of your camera, you effectively have a light sheet formed. Okay? So this is just a Gaussian beam that comes out of a spherical objective. The other option is a Bessel beam. So this is a sort of, you can think of it as a pencil-shaped beam that shouldn't diffract. That would be an ideal Bessel beam. We can't make ideal Bessel beams because it requires an infinite amount of energy. So what you actually have is this thin pencil-shaped core surrounded by a number of lower intensity side lobes. And once again, we can scan that up and down very rapidly as well to make a light sheet. The other option is you can do this with 2-photon excitation. And then you only have enough photon density in this actual pencil-shaped inner core of the Bessel beam to get excitation. And then you don't have to worry about these side lobes causing excitation in your sample. So you get a little thinner, effective Bessel beam. And then the final one that isn't shown here is a lattice Bessel beam. So you may have heard of the lattice light sheet microscope from Eric Betzig's group. And essentially what that is a number of Bessel beams that are all lined up in a row. And there's actually some interference in between them to cause some deconstructive interference, and that actually removes these little side lobes or these extra circles and gives you a nice thinner effective Bessel beam. Similar to the 2-photon one. All right, so how are these actually made? So the cylindrical lens, like I said, it's a lens that looks like this, and essentially what you're doing is just focusing the light in one dimension. So we have our light coming in here, and it's just being focused on this y-axis. So you see these arrows get a little bit shorter as the light wave progresses forward, whereas in the x dimension, it's staying consistent. Okay? So that's what's forming the light sheet with the cylindrical lens. Our scanned Gaussian beam, so that Gaussian beam that comes out of your confocal sort of has this hourglass shape to it. There's a portion of that in the middle that is fairly consistent. It's not perfectly flat. Depending on what your numerical aperture is here, the lower your numerical aperture, the more this curve sort of flattens out. And you can use the middle part of that beam waist as an effective light sheet as long as you scan it up and down really quickly. Bessel beams can be formed using a lens group known as an axicon. So that's this funny shaped lens right here. And what that does is it focuses some light from the top and from the bottom. You get some constructive interference here, and you get a pseudo Bessel beam formed right here in the focal point of this lens. And the other way to do it is to put, it looks very much like a phase ring, but essentially you put this in your light path so that you're only projecting where this black portion is where your excitation light is going through. The white part is blocking your laser light. So you just have this ring that's being projected into your microscope. Once that goes into the back focal plane of an objective, the thinner you focus this little ring of light into the back focal plane of an objective, the thinner Bessel beam that you get coming out the other side. And then that can, again, be scanned up and down to form a light sheet. The one little dirty secret about light sheet is there's this big trade-off in X versus Z. And what I mean by this is that the longer you make your light sheet, the thicker that light sheet has to become. Okay? So if you want a really high resolution light sheet with a really, really thin light sheet, you're going to get excellent Z resolution. You're not going to be able to project it across a very long distance. Okay? So this is an example of it here. This is a Gaussian beam formed with a 1-photon process. This is one formed with a 2-photon process. This one is thinner, so you're going to get a higher resolution in Z. But obviously, you can see you can only project it across this very short distance, whereas the 1-photon one can be projected over double, maybe 3 times the sample size. So this is maybe illustrated a little bit better here. So on the top, we have an image of a mouse brain that was imaged with a very long, thick light sheet that was formed with a cylindrical lens. So this is in the ultramicroscope design. And you can see we're able to image this entire mouse brain, but the detail here, you're definitely not getting subcellular resolution, and it's probably even going to be difficult to see single cells at this point. But you do get a great image of the overall structure of that sample. Down here, this is out of Betzig's first Bessel beam paper, and this is imaging just a cell with a 2-photon Bessel beam. So it's nice and short, very narrow, and you can see the unbelievable detail that you see of this cell moving and the ruffles forming on the membrane, just because you have an incredibly high resolution in a very, very thin light sheet. But you only can cover a single cell. All right? So this is the trade-off that you always have to live with light sheet. So I told you I'd come back to these different varieties of light sheet. I just wanted to introduce you to that resolution concept first before we talked about these. So if we look first at these beam scanning designs, there's quite a number of these out there now. So I talked a little bit about the lattice light sheet. So this is an Eric Betzig design. Your sample sits down here in this little chamber that's filled with water. And you have 2 objectives that dip right down in there. These are high numerical aperture objectives, so they can focus that light sheet very nice and thin. But obviously, you can see there's a lot of steric hindrance here. If you wanted to put an entire mouse brain in there, it's just not going to happen. Okay? Hari Shroff's lab at NIH came up with their dye-spim design, which is very similar, just 2 objectives that are coming in at a 90-degree angle. Your sample sits in the little water bath down here. In his design, he essentially has 2 arms coming out here that both send in excitation light, and they both have filters and cameras on either arm so that you can image on both arms as well. And essentially, you get an isotropic point spread function, so you get a nice the same resolution in X, Y, and Z, okay, out of this system. There's a couple single objective designs out there as well. So this was out of the Sibarta lab in Paris. It's a single objective design. So what they're doing is just taking a Gaussian beam that's just coming out of one side of this objective, and they just scan it back and forth from the front to the back really fast to form an effective light sheet. Sorry, actually from side to side here. And they send that up into their sample, and on their microscope slide on their sample, they actually have a little mirror that sits here, and that light bounces off that mirror and comes across through their sample, and that forms a light sheet. Okay? And this is actually the design that Leica uses in their commercial implementation of light sheet. Down here we have the SCAPE design out of the Hillman group at Columbia. And in this situation, what they're doing is they're still using a cylindrical lens to form a light sheet, but that projects into the objective just onto one side of the back focal plane so that it gets focused into your sample, and they can actually sort of tilt this light sheet so that it slides through your sample right here. And they've actually changed this design a little bit where they actually put a dichroic mirror in here so that instead of just collecting the light from one half of the objective, they actually collect all of the light. So they get a little higher resolution and send it over to a camera. So this is just a little light sheet that comes out of one side of the objective, walks across your sample. They collect that fluorescence onto a camera and build up a 3D image. Again, single objective, so it's a little less steric hindrance, but this light sheet can't be projected over that long a distance while maintaining its properties of thickness and length. Same with this design up here. Okay, so again, these give you really good resolution, but unfortunately, you're restricted to a pretty small sample size. The Multiview design came out of Ernst Stelzner's group, and a lot of his postdocs and grad students who've gone on to further labs and have furthered the design. I think Phil Keller at Genelia Farm has really pushed the technology, and Jan Huskins over at MPI in Dresden. And the idea here is you use two relatively, well, essentially you have a cylindrical lens further back in the microscope that forms your light sheet. You use some low NA spherical objectives just to focus that a little bit more into your sample. And you make this nice thin light sheet that you can then move your sample back and forth through while you collect fluorescence on this imaging objective at the back. The neat thing here is your sample is no longer sitting in between a cover slip and a microscope slide. It's actually suspended in this column. So this is a little glass capillary, and here's the sample sitting in a little column of agarose in front of the light sheet. And what you can do here is you can actually rotate that sample now and image it from multiple angles. Yeah. So I say this gives you a medium resolution. It's not as high a resolution as the really thin light sheets. Usually this light sheet's sitting in the 5 to 10 micron thickness range. If you have a really large sample, you're going to need to tile. You're not going to get the entire sample in a single field of view. And so that combination of some pretty good resolution and tiling also leads to large file sizes. So then the ultramicroscope design is essentially a stereo or zoom microscope body with some additional optics in the bottom to form a light sheet coming through your sample. Again, this is usually done with cylindrical lenses because you can make a nice long light sheet, which you need to cover the entire field of view of a dissecting objective that has a nice large field of view. So this was out of Hans Dott's group in Vienna. And here you can actually, if you're using a low enough magnification objective up here, there's no need to tile, which leads to a very small file size. Unfortunately, that means you have a very low resolution of your sample as well, so it's very difficult to even get cellular resolution unless you start zooming in and tiling again. The other nice advantage to this system is it's compatible with a lot of solvents that are now used for tissue clearing, which we'll talk about a little bit later, because you're using an air objective that's actually not having to dip into that solvent. Okay. So the other thing to keep in mind, besides that relationship between X and Z, is the relationship between resolution and maximal sample size. So the higher the resolution of your light sheet system, the smaller the sample is going to be that you can get into it. Okay? So if we're looking at that stereo or zoom design, we can get a really, really large sample in there, but our resolution is going to be pretty poor. Okay? Once we get out to beam scanning designs like a lattice light sheet, we have incredibly high resolution, but unfortunately we can only get a really small sample under there. Okay? So I think that's really the take-home message between these different designs. So what kind of samples can we actually look at in one of these microscopes? So I'm just going to take you through a few of the examples of what we've been doing with ours. I think we've tried to cover most of the possibilities, but I'll talk to you a little bit about small organisms and developmental biology. Then we'll talk about 3D cell culture and organoids or spheroids, some tissue explants, and some clarified tissue. So this is a list of everything we've thrown in ours so far. We mainly look at zebrafish. We have a lot of zebrafish labs in close proximity to us, so that's our primary sample on the developmental side. But we've also taken a look at Drosophila, some C. elegans, some crickets, some salamanders, some mice, and small aquatic organisms like this juvenile eggcorn worm that one of the labs digs up out on the coast out by the marine biology labs. So this is, we're using the Zeiss system, so we're using the Multiview type design system, and this is what it was primarily designed for, was developmental biology. So the sample on the right here is just a really early zebrafish embryo. So this is imaged just from a single angle. This wasn't actually even a multi-view image. And this was imaged every 30 seconds for about 20 minutes. And in the zoom in here, you can sort of get an idea of what the resolution of this is. If you look at, this is just an H2B TD tomato construct so that you can see histones. You can actually see some of these cells, the DNA condensing and dividing. So it is a pretty good resolution system. Obviously not quite what you'd get with confocal, but pretty close. And this is just depth coded. So the cells at the back are red and the cells closer to the objective are blue. But this brings me to a little bit of a sidetrack. So I told you we can rotate our sample here in a multi-view microscope, but I didn't really tell you why we would want to do that. And this really comes into play with developmental biology. So what you have is you have a sample that's sitting there. It's alive, so obviously we're not going to clear that sample. It means there's biological tissue there and there's going to be a lot of light scatter happening. So we'll talk about a bit about that with clearing after the break. But this is a good example of what's going to happen there. So this is a zebrafish embryo that's sitting here. It occupies this entire space. And this is what happens if you send in your illumination light, your excitation light, just from one side. Okay, so just think of it as only coming in through this objective on this side. And then we've got our detection objective up at the top here. So that illumination light is coming in, and it can only penetrate about halfway through the sample. So in general, you're getting maybe up to 200 micron penetration. Zebrafish are usually pretty clear, so you get a little deeper into them than you would into human tissue or mouse tissue. So this entire zebrafish is probably about 400 to 500 microns in size. So you can see that illumination light is only penetrating about halfway through our sample. So we're not getting excitation of this side, this half of our sample over here. Now, to the same extent, we're not able to see the bottom of our sample either. So this excitation light is exciting all of the fluorophore that's down here, but we're not seeing that either. And the reason is that the light coming from this lower half is being scattered out of the sample before it gets to the imaging objective up here. So effectively, we're only able to image 1/4 of our sample. All right? So there's a couple ways around this. The one obvious way is just to send in our excitation light from the other side as well. So if we had another illumination beam coming in from this side, then we'd be able to see the entire top half of the sample, but we'd still be missing the bottom half. So with this sample prep, when we have our sample just sitting in this column of agarose, we're actually able to just rotate this. And now what you can see is if you rotate this from 6 different angles and take those different images and fuse them back together, we can now see the entire zebrafish. Okay? The other really interesting thing about this is the more rotations that you do, the better or the more you improve your point spread function in the z dimension. And so this is just a simulation of some data imaged with between 1 and 18 different views. And what you can see is that the more views you add in here, the higher the resolution you produce in your final image once you reconstruct all of those views on top of each other. Okay? And just along the bottom here, this is an example of the effective point spread function as it becomes more isotropic and more circular as you add in more views. So to give you a real-world example of this, this is a salamander embryo, just the head of the salamander embryo that we imaged. It was stained with phalloidin, so we're just seeing all the musculature here in the head. And what you can see is we've got a good image of the bottom here, and we can sort of start to see up the one side here, but we're missing the entire top of the head and the right side over here. And so this is just what happens if you just take a single view of that data set. Down at the bottom here, we actually did 6 different angles and reconstructed those all on top of each other. And now you can see we have the entire data set totally complete all the way around the embryo. These little green dots floating around in the outside, those are fluorescent beads, and those are placed in the agarose outside the sample. And that's what's used to overlay all of those different angles onto one another. So this is done all computationally afterwards. They can be removed. It's a bit of a painstaking process. The easier way to do it is just to, so these were actually green beads with phalloidin 488, so green beads, green fluorescence, that's why they're all there. The easier way to do this is to say do red beads with a green sample, and then you don't have to worry about seeing them. Or using smaller beads that are actually dimmer than your sample so that you don't actually see the beads and you just see the sample once you reconstruct it. All right. The other thing that we're able to do is further improve this resolution by deconvolution. So this is a pretty large sample. So this was imaged with that 5x objective, the really low numerical aperture that I was showing you earlier. And so there is still a bit of blur there, a bit of out of focus light. We can run a deconvolution algorithm on this, reassign some of that blurred light back to where it should be. And now what you see is we have much more detail and much higher contrast. So the other thing that I just want to show you here, and it's a little easier to see it when it's spinning, but these beads, like I said, act as point spread functions. So you can see they have a very long, almost cylindrical shape. Once you do the multi-view, these cylinders are getting a little tighter and a little smaller and going back to spherical. And then finally, with this deconvolved image, they actually come back to looking like points. So it's a good sort of quality control mechanism for the processing that you're doing at these multiple steps. Okay, so the next type of sample that we've been looking at are 3D cell cultures or organoids or spheroids. So this one right here is a sample of, this is alveolar tissue, so stem cells that were differentiated into alveolar tissue or lung tissue. And this is just 3D cell culture sitting in Matrigel. So there's nothing too special about this. Obviously, because it's alveolar tissue, there's a lot of air pockets in there. There's a lot of empty space. This is something that's very easy to image in the Lightsheet microscope. Spheroids and organoids, on the other hand, are these really incredibly dense pockets of cells. It's actually quite shocking when you try to image them, how dense they are and how badly they scatter tissue. So when we're dealing with organoids, if they also come in all kinds of different sizes, so if you have something that's less than 100 microns in diameter, you can usually get a really good image with it on the light sheet. Just popping it in, you have to do multiview so that you image it from multiple angles. But sort of if it's in that 100, 150 micron range, you can get a good image off of it. Once they start to get bigger than that, all you're going to be able to image is sort of the outer 100 microns on the outside. So what you get is this nice detailed 100 micron 3D image with a big empty centered core. So once they get beyond that size, you actually have to go into tissue clearing to be able to image them well. But we have had a lot of success with that as well. And I'll talk about that. I'll talk about tissue clearing a little bit later. And then this is just an example of a day E9.5 mouse embryo kidney. So this was just stained. The green spots are a stem cell population in the uteric buds, and then the red is in the actual tubules of the kidney. So this, again, was an uncleared sample. And we were, sorry, that doesn't look so great on the projector here, but this was just an uncleared tissue. This wasn't imaged with multiview either. But with Multiview, you probably could have got a better reconstruction of the entire kidney. But you can see on the inside, again, you sort of have this hollow inner shell just due to the light scatter. So it's something that would have definitely benefited from clearing as well. So the other big thing that we've been imaging on the system is cleared tissue. So tissue clearing is something that's really jumped off recently. This is a table out of our recent review, and this is only part of it. It still continues on the other page. So there's a vast, vast number of these techniques out there, and this came out in the summer, and it could already be revised now. It's kind of crazy how popular these techniques have become and how broadly accepted they are now. So this is just a quick example of a couple samples that we had. So this is the one that I showed you earlier from the Urtuk group, or the 3-disco group. These are a couple that we did using the clarity technique. So this is just a subset of neurons in the brain labeled with GFP. And that's just another 3D reconstruction of a different brain that we did so that you can see it. So we'll talk more about tissue clearing after the break, but I just wanted to give you a couple examples of how it's done and what it looks like. So just before we take a break, I'm just going to talk a little bit about data handling. So this is that cleared brain that I just showed you on the last slide. And I dug through my files, and I actually found this. So this right here is a 12 gigabyte file, which doesn't seem that bad. But the other thing is that this was downsampled 4 by 4 times. So for every 16 pixel square, that became 1 pixel into this image. So what that means is that this is actually only 6% of the original file size, which was 200 gigabytes. And that's just a single color image, or just a single color that we're showing you here. It was actually a 2-color image. So there's 400 gigabytes. It was also tiled with overlap between the tiles. So that's after those tiles have been fused together, and you've blended a lot of the data at the edges there. So it was even bigger than 500 gigabytes. So these entire brain images usually end up being somewhere between 500 gigabytes and a terabyte to start with. All right. So I showed you this graph before in that as your resolution increases, your maximal size, sample size decreases. But I'm going to throw in one more data set here, and this is your data volume. So obviously, increasing resolution gives you increasing data size, and increasing sample size also gives you an increase in data size. So what that means is that the Multiview design microscopes, like the Lightsheet that we're using, the Zeiss product, gives you the largest data set out of the different varieties of microscopes. Okay? So with a stereo to zoom design, you might be dealing with 10 gigabytes of data. Same sort of with this beam scanning one, depending on how many time points you're doing. Obviously, this is just assuming one time point. Whereas here, you're going to be dealing with hundreds of gigabytes, maybe a terabyte of data. All right. So just to go back to the image of a brain, I don't know why that disappeared. So this is how we acquire it. So each one of these tiles is going back onto an SCMOS camera. So there's approximately 2,000 by 2,000 pixels there. Each field of view is approximately 2 millimeters by 2 millimeters. So each one of these tiles for a single plane is about 8 megabytes. We generally take about 40 tiles to cover an entire brain. So 8 megabytes times 40 tiles, you're at 320 megabytes now for a single plane. Do that in 2 colors, now you're at 640 megabytes. So you're pretty much at about 1 gigabyte for every 2 Z-planes that you do. It takes about 1,800 Z-planes to get through an entire brain. So that's why I say you're coming out with this file size in the 500 to a terabyte range. All right? So it's a little intimidating. Even more intimidating, let's do a multi-view time-lapse image. So now we're doing about 250 Z slices through our zebrafish embryo. Each one of those is 8 megabytes. We have 2 cameras on the system, so let's collect this in 2 colors. So there we have 4 gigabytes per Z slice. right? 4 gigabytes times 30 stacks per hour, so we're acquiring these every 2 minutes, times 12 hours, now we're at 1.4 terabytes. Seems pretty big, but then let's remember we're doing a multi-view, so we then have to multiply that by 3, and now we're at 4.2 terabytes, right? This is 12 hours. You could do this for 24 or 48. So you can see we're dealing with some pretty massive data sizes here. So when our system originally came in, so we've had a Lightsheet for about 3 years now. Our original system came in, all the systems come with 2 computers. So you have one computer that hooks up to the actual microscope. This is what the microscope looks like, big laser printer. And you have a computer that's hooked directly to that is your acquisition computer that drives the microscope. That computer does not store any of the data. It's connected by a 10 gigabit optical fiber that goes out to a separate processing station. So the original one that we had was 8 terabytes in size. We now have, with our newer system, a 30 terabyte size, which is the standard one that comes with the system. And from there, what we were doing was just dumping all of this data onto a whole bunch of external hard drives. There was actually one lab that ordered 150 of these in bulk, and they still have a filing cabinet sitting in the lab full of these with basically 1 or 2 data sets on each hard drive. So we muddled our way through that for a little while and then decided that maybe wasn't the best situation. So now what we have is we have our Lightsheet acquisition computer as well as a number of our other high data output systems have been rewired throughout the entire facility with 10 gigabit optical fiber. So what this means is we can move data from one computer to another really quickly. So these are our microscopes that sit on this fiber network. And then we also have 2 processing computers that sit on the fiber network. And all of these are connected to our expandable storage space that sits at a server farm in downtown Boston. So we currently have, actually this should say we're up to 200 terabytes now of dedicated space for our facility. And this is just in an expandable server farm. So for $8,000, we can buy another 50 terabytes of space whenever we need it. And it's just housed in that server farm. A number of the other labs, so including that lab that I told you that had the filing cabinet full of hard drives, they have now also bought their individual space sort of in this same server farm environment. So I have no idea how much additional space the individual labs have, but it's probably fairly substantial as well. And all of this data lives on the Harvard Odyssey cluster computer. So what it means is for processing really expansive data sets, it can just be dumped onto the cluster computer for really fast processing, which is a nice advantage to where we are. So obviously, this isn't possible for everybody. So the things that I find are the really most important is figure out where your data flow is going to be, which computers it's going to move through, and make sure you have the fastest possible connection between those computers. Okay? If it's only 2 or 3 computers, it's not a huge expense to make that happen. The other thing is this idea of expandable storage space. It's actually pretty cheap right now. So like what I said, $8,000 for 50 terabytes. Although we do that at our server farm, that's something you can do in a closet in a lab. It's definitely possible. So you don't need some sort of crazy network where you're sending stuff downtown. But the key components are that you have that really fast transfer rate in between all of your different systems, and then you have some expandable storage. Because no matter how much you think is enough, it's not going to be. Okay. So then the next thing you have to do is somehow process this data and deal with it, right? And so this is generally a 3-step process. So the step 1 is with all data processing, we usually have to convert from some sort of proprietary file type that comes off the microscope into something we can actually use. The second step is if we're doing a large tiled image of a large cleared tissue, we have to stitch those tiles together. Or if we're doing multi-view, we have to reconstruct all of those multi-views on top of one another. And then finally, we're doing some 3D rendering or some quantitation of that data, right? So there's some images, so there are multiple methods to do this. There's the expensive way, there's the free way, there's the fast way, there's the slow way. This is what I've found to work best. So as far as converting from this proprietary file type, the free way is to do it with Fiji. So there's actually some macros that you can download from our facility's website that will just convert your file, the CZI file that comes off the microscope, into a bunch of TIFF stacks. And this takes, depending on the size of the data, maybe 30 minutes, maybe a couple hours. It just all depends on how big that file type is. The expensive way to do it is with this Arivis software. So this is a small, now very much growing startup company out of Germany. It's a product called Arivis 4D Vision. It's actually, you can get it packaged with the microscope, which I highly recommend. And this is a software program that, unlike Ameris or Velocity, it's not limited by data size. So they've tested it with file sizes up to 10 terabytes, and it's been fine. I could actually run it on my laptop here with a 10 terabyte data set. I could 3D render it and show it to you. It doesn't require a lot of computing power. It's really a pretty phenomenal piece of computer science. So it can also do this conversion, okay? And it's going to take basically the same amount of time as Fiji. So the next thing, if we're, let's look at stitching tiles, first of all. This can be done in Fiji or in Arivis. I'd say this is one of the greatest powers of Arivis, is this can be done in a couple hours. It's very easy, it's very user-friendly. Fiji can also do this. It's going to take a lot longer. Fiji likes to load everything into RAM. Obviously, you don't have 10 terabytes of RAM on your computer. So you have to do it in virtual stacks where Fiji is just loading in a single image at a time. And that takes a long time. So you can do the stitching in Fiji. It's going to take overnight. And it's probably going to take a few attempts at it before you get the parameters right. Whereas the Arivis software is very interactive, and you pretty much get an idea of whether it's going to work right off the bat before you actually hit go and wait a couple hours for the final product. Yep. There's another program called TerraStitcher, which I haven't used, but some of the people using our facility have used. It's, again, open source and free, and apparently it's pretty quick and doesn't require a lot of computing resources. But it does have, I don't know if they've changed it now. Hopefully they've made it a little bit better, but it has this horrific data structure that your data has to sit in a number of folders that have to have specific names and a whole bunch of subdirectories that have to be in a specific order for it to actually work. So it's usually, although it takes hours to do it, it often takes hours to set up your data into the right directories and subdirectories. Okay, so now if you're doing multi-view reconstructions, this can only be done in either Fiji or Zen, which is a software that comes from Zeiss. Sorry, I didn't put Zen there. So Zen has a really nice manual alignment. So basically what you do is you get a maximum intensity projection of all of your different views, and you just sit there and line those up on top of one another. And then you press go, and it reconstructs them all on top of each other. It's nice because you're guaranteed to always get a reconstruction. It's not as accurate as the other methods where you're using the fluorescent beads to overlay everything. But you can usually get something pretty good. And it takes, depending on the size of your data set, maybe an hour or two to do that manual alignment. And then the computer is probably going to crunch the data overnight, or maybe even a couple days if you had a really long time lapse. All right? The other option is to do it with bead-based recognition. So I showed you those fluorescent beads in the agros around the sample. And this can be done on Fiji or in Zen as well. Sorry guys, I highly recommend Fiji and not Zen. Zen is a little too particular in what it accepts as a bead. And it usually throws out a lot of your beads, and it also takes a pretty long time. So Stefan Preibisch, who used to be at Genelia Farm, he now has his own group in Berlin. He's written some phenomenal Fiji plugins that will allow you to process this multi-view. If you just have a single time point for views, it maybe takes half an hour. If you have a really long time-lapse data set, it's going to take overnight. We've actually, and other people have actually altered the code or worked on the code to put it on a cluster environment as well. So then you can do it in a couple hours on a cluster environment. So then finally, you want to 3D render this and quantify it. For 3D rendering, especially if you've used the Fiji plugins for the Multiview, there's something called the Big Data Viewer out of Pavel Tomachik's lab in Dresden. That's another plugin for ImageJ Fiji. And it allows you to visualize the data. It doesn't really allow much for exporting that visualization or annotation. I haven't looked at it in a while. Maybe they've added that in. But it does at least allow you to 3D render it and view it. There's another program called VAA 3D, which I haven't used, but a lot of people that use our facility really like it. Again, it's open source and free. And then finally, there's Arivis, which does a fantastic job of the 3D rendering. Arivis is still a new company, so they sort of haven't got to the same level of quantification ability as, say, Ameris does. But Ameris just isn't an option anymore because it can't deal with files this large. So as far as quantitation studies, we've done that primarily in Fiji or ImageJ up until this point. So just to give you an example of kind of what this looks like from start to finish, this is a great paper out of the Yoshida lab that just came out this year. And essentially what they did was they looked at nearly 80 mouse brains that they'd cleared. And what they were doing was they were injecting GFP rabies reporter virus into various regions of the brain. And this rabies virus will basically label any neuron that's connected to a dopaminergic neuron. So we're looking for innervation into dopaminergic neurons. And depending on, obviously, where they inject it, they can see where the neurons from that region of the brain, which dopaminergic neurons they connect to. So essentially their process was they did their injections, waited a couple weeks, and then went through the Clarity protocol, which we'll talk about a little bit later, but in their version here is taking about 10 days to clear the brain. And then on day 11, they did their Lightsheet microscopy. This takes about 4 hours to image an entire brain. And then finally on day 12, they did their data analysis. So a 12-day process, start to finish, for each brain. All right? So what they're doing here, this is an image of the brain. And you can't quite make it through an entire mouse brain from one side in the Lightsheet microscope. So what we do is image it halfway, spin it 180 degrees, and then image it from the other side. All right? And this is just representative sections all the way through that brain. So then what they were interested in was just finding cell bodies and determining which region of the brain they were in. So what they had was some segmentation software that they were able to take an image like this and just segment out the nuclei. And they actually had to have different algorithms for different regions of the brain because there's different levels of background. Now this is just the segmented data over here. So you can see where the little cell bodies popped out of this original image. And then what they did was they overlaid that onto an atlas of a mouse brain and transformed it so that it fit perfectly on that atlas. And then they counted how many cell bodies they had in each region of the brain. So I think what's impressive about this is they started out with over 80 mouse brains that they cleared. 77 of them were clear enough to actually be imaged on the microscope. And this represents over 150 terabytes of data. But in the end, in the paper, it turns out to be one graph that's a few kilobytes in size. Here's the picture that we've seen 1,000 times already, just reminding you of that we're not dealing with cover slips and microscope slides anymore. What we're dealing with is some sort of cylindrical tube that's holding our sample. And there's a few options here. So this is just a couple pictures of the inside of the microscope so you can get an idea of what's going on inside here. So these two little black objectives on the sides here, those are the Lightsheet objectives that are projecting in our excitation light. And they're going to be focusing it right here in the middle of this objective, which is our imaging objective that's collecting the light at the back. So this is the 5x objective. So this is an air objective that sits outside of the sample chamber. There's also some water dipping objectives that you can get that go in here, and they actually fit right into the chamber and form sort of the fourth wall of the chamber. This is the imaging chamber over on this side. And so what you can see is just a big piece of metal with 4 cover slips on each side. Our excitation lights coming in through the sides, imaging objective at the back. Down here we have a little Peltier heater that can heat up this chamber if you're doing some live cell imaging. And then these 2 little lines here are just water circulation to actually cool that down if it gets too warm. And this is just a little power supply for that Peltier cooler. And then over here we have a little inlet port. This is where we connect a syringe via an IV extension line, and we use that to fill the chamber with our imaging solution, which is usually just whatever buffer your sample is happy in, or the clearing solution that you've used to clear your sample. All right. This is just a little emergency overflow for when you fill it up too high, and then that can drain out the bottom of the microscope and hopefully not damage anything. There's also a little temperature sensor here that will actually sit right inside this chamber when you're doing live cell imaging as well. Okay, so our 2 main tools for mounting samples are these glass capillaries. So this is traditionally what's used, or a 1 ml syringe. Okay, these glass capillaries come in 4 different sizes. They range everywhere from 700 microns up to about 2 millimeters. So those are for our smaller samples. They fit in this big metal holder that then plugs into the microscope. And as well, there's a secondary holder that can hold these 1 ml syringes as well. So what I'm going to do is I'm just going to start from small to big and show you how you mount these different types of samples. So the small living samples, these are just for short-term imaging. This is the classical mounting technique. So you take a glass capillary. You put your sample in some 1% low melt point agarose that's been melted. So it's probably sitting at about 40 degrees Celsius. You put your sample into that agarose, suck it all up into this capillary. The capillaries come with these little plungers, so it acts just like a syringe. Pull it up in there, let that agarose harden, and then put it in the microscope, push the plunger out so that your agarose column comes out the bottom. You don't want to image through the glass here. It's not optical glass. You're going to get a terrible image. So you push your sample at the bottom, so just this little zebrafish here. Right. So that works for short-term imaging. If you're going to do long-term imaging, you run into a problem when that zebrafish is sitting in 1.5% agarose. So you can see this was grown for 24 hours. This one was grown in the agarose. This one was just left swimming around in water. And you can see there's some major developmental defects here. This guy just isn't growing as well as he should be. So if you want to do some long-term imaging, longer than a couple hours, you actually have to modify this mounting a little bit. So in this situation, instead of a glass capillary, what we use is a tubing. This is called FEP tubing. It's basically Teflon tubing. And what it does is it has the exact same refractive index as water. So once you dunk it in that sample chamber and you have it filled with water or PBS, this tube is invisible to the microscope. It just doesn't see it. All right? And then what we do is we put our little fish in 0.1% agarose, so a much lower concentration of agarose, and then just stick a little 2% agarose plug at the bottom for him to sit on the bottom of the tube there. And then once he's in this, there's enough agarose, this 0.1%, that he's not going to flop around and jump around, but he's still able to grow and develop properly. And so this works for longer term imaging. This is like a couple days. 24 hours, 48 hours. So there's a couple things not to forget. So one of the big things is if you do have something that's going to be moving around, you need some sort of anesthetic for it. So with zebrafish, it's usually tricaine, but something that's actually even better is to inject either bungarotoxin protein or the mRNA for bungarotoxin. And it just totally paralyzes the fish, but doesn't affect things like heartbeats or anything like that. It's actually a little bit better than tricaine. The other thing, if you're doing multi-view imaging, don't forget your fluorescent beads. In the end, you're trying to get about a concentration of 10 beads per every field of view. So when you have a single Z-plane up there, you want to be able to see about 10 beads. This is different for every single type of fluorescent bead you order, because they obviously all come in different dilutions. But this is just an example for some 100 nanometer beads that we have. It takes a 500,000 times dilution. We also have some 2-micron beads, and those it's like a 1,000 times dilution. So there's a really broad range, and it's just something you've got to figure out on your own. Usually we just do a bunch of different dilutions, put them in the glass capillaries, throw it in the microscope, test it, find the dilution that's right, and then mount our sample in that dilution. That's the quickest way to do it. So you're going to dilute those beads, sonicate them to break them up because they're usually in little clumps, add them to your agarose and vortex them really well before you put your sample into it. Right. So now if we have a medium-sized sample, so similar to that salamander head that I showed you earlier, it's not going to fit in one of those glass capillaries. That FEP tubing comes in a bunch of different sizes, so that's a possibility. But another option is just to take a 1 ml syringe and just cut the tip off of it so that you've got a nice wide diameter here. And then again, you just fill it with low melt point agarose, drop in your sample. So here we got a little mouse embryo. Let it harden, push it back out in the microscope, and go ahead and image. The one thing to remember here is this is now about 5 millimeters in diameter across here. The working distance of the objectives in the microscope is generally 2 millimeters for the 20x, 40x, 63x. So you're not going to be able to cover this entire sample with the high resolution objectives. You've got to go down to the 5x objective, which has about 5 millimeters. Yeah, so it can cover this entire sample. Okay. Now, if you have a big sample that isn't going to fit inside that syringe, you can directly attach it to the end of the 1 ml syringe. So again, we've cut the tip off of that 1 ml syringe, just filled it with a bunch of super glue. I prefer super glue gel. And then just dropped our little mouse embryo right on the top there. Give it about 20 seconds, it hardens, dunk it right into the microscope. So this was an embryo that was cleared with the cubic technique. There's also a couple special holders that you can get from Zeiss. But I find this works just as well, and it's pretty easy. Now, if you have a very large sample, so I showed you this image earlier of the inside of the microscope. What I didn't show you is what's going on up here. So there's another little door at the top here that you lift up, and you drop this syringe or glass capillary down into the microscope. So this is looking down from the top through that little door. And this is the actual stage that holds this sample holder. So there's 3 little magnets here that all pop onto 3 little magnets on here. This hole right here is 1 centimeter in diameter. So if your sample is bigger than that, it's not going to fit through that hole. Okay? So what we actually do in that situation is you can take a couple magnets, so just little small hobby magnets, and you can either glue that to the end of the syringe, you could glue it to the end of a capillary. Oftentimes I just drop it in the end of the syringe and clamp it in there with the plunger. And then you take another one of these magnets and you super glue it onto your sample. Okay? And then what you can do is put the syringe in the holder, drop it down into here, and before you put your chamber in, what you can do is actually just look up inside here. And what you're going to do is just take your sample with the magnet, stick it up in there, connect the 2 magnets, then put your chamber back in here, drop it down into the solution, and go ahead and image. Okay? That said, you're still pretty much limited in this design to something that's about 1.5 millimeters wide. So your sample can be up to about 2 centimeters in this dimension, but only about 1.5 centimeters this way. Okay? That's pretty much all the travel you have to go side to side while you're tiling in the microscope. And the other dimension, in the Z dimension, you can get through usually about 4 millimeters in really well cleared tissue, sometimes 4.5 if you're lucky. And then you've got to rotate it at 180 degrees and go from the other side. Okay? So again, there you're limited to just under 1 centimeter in the Z dimension. All right. So now the other thing is cleared samples. So if you have some cleared samples, the one thing you want to remember is most of the solvents that are used for solvents-based clearing are not compatible with objectives or any plastic. So this means they're not compatible with this microscope. If you put them in there, they will destroy any objective they come in contact with, they'll destroy any plastic that's in there, and end up leaking out all over throughout the microscope. The other thing that you want to remember with cleared samples is to make sure that you refractive match everything as best as possible. And the key step to this is filling the chamber with the exact same clearing solution that your sample's been sitting in. So usually we'll leave your sample sitting in 30 mils of clearing solution, put your sample in the microscope, and pour that exact same 30 mils into the microscope so that you have a perfect refractive index match throughout your sample and throughout that chamber. The other thing is some of the tissue clearing methods produce a pretty fragile sample. These can be embedded in agarose that's been dissolved in your clearing solution. And then you can harden them up a little bit harder, trim them up, and you can actually glue those right onto the magnets as well. Right. Cool. So that's basically sample prep. It's obviously, you got to start thinking it's a little bit different. It's not your microscope slide and cover slip that you're used to. But personally, I find it way easier than slides and cover slips. I'm the king of air bubbles with cover slips. I have never been able to cover slip a slide properly. So this microscope has saved my career. OK, so in the last, what do we have here? Half an hour. I'm just going to give you a quick introduction to the various clearing techniques. So I think I've shown you why this is necessary already to this point, but just to give you a quick reminder, this is an image out of one of Eric Betzig's papers from a couple years ago. And this is looking at neuronal tissue in a zebrafish, just with a standard confocal microscope. And what you can see is as you go deeper and deeper into this tissue, your image begins to become more and more blurred. All right? So in general, with a confocal microscope, with a one-photon excitation process, a continuous wave laser, you're limited to, at maximum, about 200 microns penetration. Okay? If we go to a two-photon excitation process where we're using infrared light to get a little bit deeper into our sample, we can get up closer to 1 millimeter. But these are pretty much hard limits for biological tissue. All right? Just to show you the rest of this figure, Betzig's group actually put what's known as adaptive optics or deformable mirrors in their microscope, and then you can actually play with the wavefront of the light as it's going in or coming out of the microscope. And they were able to clean up these images. Again, this still has a limit to it. It's not going to get you to the point where you can image all the way through 5 millimeters through a non-cleared brain. It's not going to happen. So a couple things that you have to keep in mind with tissue clearing. So it's not necessarily a method to remove autofluorescence. If you look at the different techniques that are out there, some of them include a step that will help with autofluorescence. But in general, if you have autofluorescence in your tissue, it's still going to be there after you've cleared your sample. Okay? It's also not necessarily a method to remove or reduce absorption. So if you have a lot of hemoglobin, myoglobin, melanin, that absorption, those absorbing molecules are still going to be there afterwards, and that's still going to pose a problem. All right? What it is it's a method to homogenize light scatter throughout a sample. We often talk about like biological tissue being scattering. The truth is that everything scatters light. Whenever light hits matter, it scatters. The difference between a glass of water and a glass of milk is how homogeneous that light scatter is. Okay? In a glass of water, that light scatter is very homogeneous, and there's a lot of deconstructive interference in the lateral or reverse light paths to the initial light path. So in the lateral or reverse directions. But in milk, there isn't that deconstructive interference. Light just goes everywhere. And so the easiest way to think of this is whenever you have a mismatch in refractive index, so for example, in milk, it's when you're going between lipid and water. Whenever you have a mismatch, you have an inhomogeneous scatter of light, and you get a cloudy sample. So what tissue clearing is trying to do is match that refractive index all the way throughout our sample so that you get a very homogeneous light scatter. You get all that deconstructive interference in the lateral and reverse directions, and your light is just constructively interfering with itself and producing a straight path straight through your sample, and you end up with a nice clear tissue. So what are our scattering centers in biological tissue? So we have a lot of intracellular fluid and extracellular fluid, which is made up of water and salts and has a refractive index a little above water, around 1.35. The main scattering centers in biological tissue are lipids. And not so much the plasma membrane. It's pretty thin. There isn't a lot of time for that light scatter to happen. More importantly are mitochondria and Golgi, because you have a double membrane layer that's then in ruffles over on top of each other, layered on top of each other. There's a really big refractive index mismatch there between the cytoplasm around it. And so this has a refractive index around 1.45, 1.47. Proteins throughout your sample, especially hydrophobic proteins, these also have a pretty high refractive index, and structural proteins like collagen or elastin. This can be up over 1.5 when they're dehydrated, and in the range of 1.47 while they're still hydrated. So again, lipids are the biggest one, but proteins definitely contribute as well. So the tissue clearing methods have been divided into two main categories. So organic solvent-based clearing and then aqueous-based clearing. And we've further tried to categorize the aqueous-based techniques into simple immersion, hyperhydration, and hydrogel embedding. So I just want to walk you through these and just sort of give you an idea of the molecular mechanism behind them and how they're doing what they do. So solvent-based clearing, much like Lightsheet, is not a new method. Again, it came out in the early 1900s. So Werner Spattenholz in Leipzig, Germany, was developing these techniques to clarify really big anatomical specimens. So he taught anatomy at the local university, made some great anatomy textbooks, and he came up with these ways to, say, clear an entire throat so that you could see the vasculature better. So he had a technique that was pretty harsh. It damaged a lot of the tissue on the outside. So it's not something that would work for the size of samples that we're interested in imaging in a microscope today. So since Spattenholz, more recently, there's been a lot of refinements of his technique. So we've gone from BAB to 3-disco to iDisco now is the latest one. And at Neuroscience this year, they presented U-disco, which was ultimate disco, and iDisco plus. So like I said, this changes monthly. But the concept behind all of these is essentially the same. The first thing you do is take your sample through graded alcohol dehydration, usually with methanol. And what this is doing is removing the water from your sample and a little bit of lipid as well. And then finally, you put on one clearing solvent, maybe a couple clearing solvents. This gets rid of some more lipid. Refractive index matches the tissue to the solvent around it and turns it clear. All right. So it's a really simple process. It's very robust. What you've done is you've removed all that intracellular and extracellular fluid. You've removed the lipid. You're just left with some dehydrated protein and your refractive index matching to about 1.5. So these are a number of the molecules that have been used in this process, and you can see they all have nice high refractive indices around 1.5. All right. So that's solvent-based clearing. Aqueous-based clearing, we'll take a look at simple immersion first. Simple immersion is just like the name sounds. You're just dunking your tissue in a single solution, exchanging that solution every few days, and eventually your tissue's hopefully going to turn clear. All right? So there's a number of techniques that sort of use this. So CDB is a high concentration of fructose. You can do it with a high concentration of sucrose, glycerol. Focus Clear is a commercial agent that's available. TDE is another solvent that's kind of water soluble as well. Fruit is a combination of fructose and urea, and Clear T is formamide. So essentially all you're doing here is just replacing this intracellular and extracellular fluid and trying to match as best you can everything else that's been left behind. Okay? So all of these clearing solutions all have a refractive index somewhere in this range. Hyperhydration is very similar to simple immersion. It's the same idea. You just dump a clearing reagent on your sample, exchange it every couple days, every couple weeks, and eventually your sample is going to turn clear. This is originally scale was the first technique to use this hyperhydration method. Scale S then just recently came out. There's a couple versions of cubic, clear tea and fruit all use this. And the big difference here is there's, at varying degrees, these different techniques introduce some detergent to form some micelles and get some of that lipid out of your sample. But the other thing that they use is urea or formamide, which can actually penetrate into the hydrophobic regions of protein through osmosis. That brings in water, loosens up that structure a bit, and reduces its overall refractive index. So what you're doing here is replacing that intracellular and extracellular fluid and removing the lipid. And you're also hyperhydrating your sample. So you're trying to reduce the overall refractive index of what's been left behind. And this ends up coming down to about 1.38, which matches the refractive index of these solutions. So finally, the last technique is the hydrogel embedding method. So this is Clarity, packed pars, and now Switch just came out recently as well. And these are a little more labor intensive. The first step is embedding your tissue in a hydrogel, which is acrylamide. This can either be done through perfusion or just through passive diffusion. Usually it's done through perfusion. So you perfuse your mouse with saline just to wash out all the blood, and then you perfuse it with paraformaldehyde, a hydrogel monomer, which like I said is usually acrylamide, and a temperature-sensitive crosslinker. Then your next step is to heat up this sample, and what that does is it causes this temperature-sensitive crosslinker to cross-link all of the acrylamide monomers that are in your sample and form a hydrogel. And then the next thing that you have to do is remove all of that lipid. So this is done with a SDS and boric acid solution. You can do it passively. You can just let your sample sit in it for a week or so, or you can put an electrophoretic charge across it, and this will actually pull it out much faster in a day or two. And then finally, you dump on a clearing solution, which matches the refractive index of what's left behind and gives you a nice clear sample. So this is just an example of some skeletal muscle we did. First of all, I'll just say these are 4 different pieces of tissue, so the tissue did not shrink going through this process. These are 3 different pieces of tissue that were treated differently. So this is just skeletal muscle if you just cut it out of a mouse. This is skeletal muscle of a mouse that's been perfused with PBS to wash the blood out of it. This is skeletal muscle that sat in that SDS boric acid buffer to pull out some of the lipid. And then this is the sample that's been finally put in that refractive index matching solution. All right? And this was a brainbow sample. Not necessarily brain, but it was a bow sample. So this is just labeling the neuromuscular junctions within that skeletal muscle. So this was just imaged just by confocal afterwards. So what we've done here is replace that intracellular and extracellular fluid, removed all of the lipid, and just added in a final clearing solution that adjusts the refractive index to about 1.47. The 2 key molecules that are used for this are histidines. So this is used in an open source recipe out of the packed pars paper. This is a solution called RIMS, refractive index matching solution. So this is what we usually point people to. This is ditrioxoic acid, which is the key ingredient in Focus Clear, the commercially available solution, but it's insanely expensive. It costs you about $1,000 to buy enough of it to fill the chamber in the Lightsheet. So this is about 10 times cheaper. It only costs you $100 to fill the chamber. And it can be reused a couple times. So if we just take a quick comparison of these, solvent-based clearing gives you really, really good clearing. It's really fast and reproducible. It's easy, it's economical. But there's a few problems with it. One, it's not compatible with fluorescent protein. So if you have fluorescent protein in your sample, once all of the water comes out of that sample, it denatures the chromophore of your fluorescent protein. You don't get fluorescence anymore. It's also not compatible with dipping objectives. So if you put a microscope objective into these solvents, you're going to destroy it. And finally, there are often a lot of tissue distortion. There's a lot of shrinkage with these techniques, which I think is something that's going to be addressed with iDisco Plus and UDisco. Simple immersion, the key here is your lipid is still remaining. So if you're doing any labeling with lipid dyes like dye or dye-O, you can still do that. It's also not going to quench your fluorescent proteins. But these don't give you the best clearing. There's still a lot of lipid there, so obviously it's not going to be cleared that great. Usually I don't recommend doing anything more than about 2 millimeters with one of these techniques, even though that's kind of pushing it. It's also pretty tough to work with these really viscous solutions that have a lot of sugar in them or a lot of glycerol. Hyperhydration, very similar to simple immersion. You also, unfortunately, get a really fragile sample in the end here. So you usually end up agarose embedding it afterwards so that it doesn't fall apart. Hydrogel embedding gives you a good cleared sample and good protein retention, but it is pretty technically demanding, which means it's not the most reproducible until you've had your hands on it and been doing it for a while. Okay. But I think these two, the clarity techniques and the disco techniques, are really emerging as the two options out there, the two best options. And I think that's what's going to continue to evolve. These guys just need to find the right solvent that isn't toxic and isn't going to damage your microscope, and it's going to be perfect. I think the switch technique, which is the latest evolution of clarity, also has a lot of potential as well. That said, don't feel that you have to be locked into one of these techniques. So if you look at all of these techniques, there's, the process can be put into a few different categories. So there's essentially optional pre-treatment, then you do your lipid removal, then maybe you do some immunolabeling, and then your final clearing. So we can do this. We can follow, say, the iDisco protocol, where we start with peroxide bleaching of our sample first, and then go to dichloromethane to remove our lipid. And then we go down, we do some immunolabeling, just some passive immunolabeling, just sitting in our antibody for a while. But then we want to image it in a Lightsheet microscope that's not compatible with solvents, so we put it in the RIM solution instead. Okay, so you can mix and match these. You can, say, do an amino alcohol treatment to get rid of heme at the beginning. Then you do SDS and electrophoresis to remove your lipid, much like you do with clarity. Then maybe you skip the immunolabeling because you have some fluorescent proteins in there, and then you dump it into TDE afterwards. So there's a lot of mixing and matching that can be done here that can optimize things for your sample, or even to just in general optimize these protocols further. So when it comes to imaging, there's 5 big limitations: objective working distance, refractive index mismatched optics, damaging solvents, the amount of time you're going to take to image this, and data storage. So we've talked a lot about this already, but just to point out, as far as the imaging goes on the objective side of things, this has been addressed by most of the major manufacturers now. They now all mainly supply objectives with a magnification of less than 25 times, numerical aperture over 0.9, so you get nice high resolution. So I'll have a working distance over 5 millimeters. They're dipping objectives, so again, don't use them with solvents, and they're refractive index matched to the various solutions. But if you did want to image this on a standard confocal or 2-photon, if you wanted to image an entire mouse brain, 20x magnification, the 1 micron z-step, imaging at about a frame per second, takes over 4.2 million images. It's going to take about 50 days to image it. So what you really need is a faster microscope. A Lightsheet microscope takes 3 to 4 hours. And we have now come full circle. So I'm done. I just want to say thanks to a bunch of people, a bunch of labs that have provided a lot of the samples that we've done for this, mainly Catherine Dulock's lab and the Yoshida lab. A lot of helpful discussions with Jeff, who wrote the clearing paper with me, as well as Nicolas Renier from the Tessier-Levine lab. They're the ones that came up with the iDisco protocol. Our funding comes through Harvard, NIH, and HHMI. Thank you.